So today we will discuss contact modes and dynamic modes of AFM. I've already covered constant height and constant force modes for tunneling microscopy, if you remember. So these modes work in a similar way with AFM, but we will get into a bit more details. Lateral force AFM, I mentioned a bit about this mode last time, but again today will be more details. And also we'll talk about dynamic modes or oscillatory modes, or another way to call the same parameters amplitude modulation. We'll be tapping, non-contact, and phase imaging. And also I will bring you some examples of each mode so you can understand where it can be used. So first of all, let's come back to force to distance versus distance curves. So this curve should be familiar to you already. So the what we can see here is that the mode can depend on Lennon-Jones potential, and here it shows different force regimes which correspond to interatomic forces. So you can see in this figure that the atoms are separated by a large distance on the right-hand side. And once you bring atoms closer to each other, let's say a few angstroms apart, they start to be attracted to each other. And this attraction increases until they come so close together that the electrons begin to keep away from each other electrostatically. So this will be repulsive electrostatic force. So when the distance between the atoms of the tip and the atoms on the samples reach few angstrom, it's about the length of the chemical bond. So the force will go to zero. So here you can see that the contact region, contact mode, mostly will be corresponding to this part of the curve, as when the tip is in the contact with the sample. This is semi-contact mode, and this part is when the tip does not touch the surface at all. So this is the ideal curve, and let's just think about what can happen and what can go wrong. And these are the examples of the curves when something went wrong. So for example, curve A, the slope of the right part here is not horizontal. So what is wrong here? So it can be due to laser interference or poor sample grounding. It can be due to thermal shift or any other long-range probe sample forces, right? Remember, so this is a distance, tip sample at the distance. So this means that something is going wrong in this particular mode. So to overcome these kind of issues, people usually tilt the baseline and they perform a procedure called baseline fitting. So this will straighten this line at the right hand part. On the B plot, you can see a discontinuity on the upwards slope section of this curve. So this is the place where the probe is intending into the sample, this approach curve, right? So this indicates a breakthrough of some material on the sample, or probably force-induced movement of different sample feature. So for example, when you study lipid bilayers, this feature can be used to confirm if you have a presence of a bilayer or you have only one layer of lipids. So might be an important feature that you're actually looking for. On the C plot here, you can see so-called multiple pull-off events, and they're in different size because the peaks are a bit different altitude. So this particular curve can be measured in experiment when you are interested in cell-to-cell adhesion, and this curve reflects how the receptor pairs between cells are breaking during the retraction part of the measurement. So another thing that you should note and see that the retraction curve is separate from the approach curve here. This could be due to cell properties. So cell is like a viscous object, and any like biological material actually shows this ideal elastic behavior when two curves are mimicking each other. It's not very often. So this thing is not very often for biological samples when two curves are exactly matching each other. And the last curve here, we can see quite unusual like transient event in the retraction curve. This can be happening by passing through the laser path during the measurement. So, for example, this particular curve on the slide was taken from the experiment when it was measured in a liquid medium, and the cell that was measured could be floating, or another piece of debris was passing through the laser beam, and what happened here. So the measurement was disrupted here. So in situations like this, what you can do, you just make another measurement, and that's it, because this is a clearly this is clearly not your sample that you intend to measure, some debris floating around. Okay, so again, AFM has 3 modes of operation. This is contact mode, non-contact mode, and tapping mode. So just a quick reminder from the last lecture that the contact mode is the first mode developed in AFM. It's the quickest mode, and its advantages is that it's easier to perform than dynamic AFM. It works in a high scan speeds, and it can be used when you have a very rough sample as well. Disadvantages is that you will notice lateral forces on your sample, and if you got loose particles, they are going to be moved and pushed out by the way how this tip is moving. If you have any capillary forces, you will be fighting with them because the tip of the cantilever will be forming a meniscus with a sample, and again, this can disrupt your measurements. Softer materials might be damaged because again, the tip is constantly moving along the surface. So non-contact mode has a huge advantage over the contact one, because basically due to the oscillations of the cantilever here, and it decreases tip to sample forces. Meanwhile, it's still maintaining the high resolution. And the tapping mode is the mode that takes all the best from contact mode and non-contact mode. So it can measure quite fragile samples, and in terms of the speed, it's kind of a bit slower than contact mode, but much faster than non-contact mode. So this is just a quick reminder. Okay, so now let's go here. And so in contact mode, you have deflection of the cantilever, and it will reflect a repulsive force on the tip. So repulsion force F, that is or interaction force, that is shown here. It will be related to the cantilever deflection, that is value x here, and it will follow a Hooke's law, where K is the spring constant of the cantilever. So the spring constant is very important property of the cantilever, and you should look at it before you start your experiment, because depending on the sample, depending on the mode, the spring constant it will be different. So this is the constant height mode, and the main advantage of the constant height mode is the high scanning speed, because it is restricted only by resonant frequency of the cantilever. The disadvantages of this mode is that the samples should be sufficiently smooth, and when you working with soft materials like biological samples, polymers, and so they can be destroyed by scratching, because again the tip is constantly touching the surface here. So yeah, this is the constant height mode in the group of contact modes. And here is the constant force mode. So the main advantage of the constant force mode is that it is possible to measure with very high resolution simultaneously for topography and some other characteristics, for example, friction force or spreading resistance and so on. Constant force also has some disadvantages. For example, the speed is restricted by the response time of the feedback system. And again, when you work with soft samples, they can be destroyed by scratching. Yeah, so as you can see, the idea is very similar to STM that we were discussing previously. And let's go to lateral forces mode in the group of the contact modes. So here, what we can say. So the physical basics is the following. When scanning in this mode, we detect the longitudinal and the vertical the longitudinal and the vertical deflection of the cantilever. And this mode is particularly effective for measuring the friction of the surface, because here you can see the twisting of the cantilever depending on the surface of the sample. And when you operate in this lateral force mode, it is important to be aware of the topography in general, because it can convolute with the signal. And this happens because the changes in the topography will occur will start the torsion onto the cantilever as well. So here you can see the example how the feature is actually twisting the tip here. So the left, so here, you can see that the effect of the feature of the surface. So for example, on the black side black part here is the different feature on the surface, and the friction here is a bit different in comparison with the gray area of this surface. And here we can see the contrast in the curves here, and please also note that depending on the direction of the scanning, so here we're going to the right and here we're going to the left, the twisting happens in an opposite direction. So that's why when you check the curves, they might have a bit different features, and usually when you superimpose all of them, then you can get what is actually happening here and below, the difference. So what you're interested, not the sign of the curve plus or minus, but you're interested in the difference that occurs between different features of the sample. So where we can use lateral force measurement, it is important for characterizing semiconductors, polymers, films, and so on. Different chemical characterization of the surface as well, frictional characteristics as I've already mentioned. And here you can see the advantages and disadvantages. So again, for the disadvantages, the same thing as for all contact modes, that soft samples can be destroyed, and the cantilever also does not last very long as well, because it is in the constant contact with the sample. I would like to move to the examples of the applications for the contact mode. So this is an example of the work conducted with self-assembled monolayers, and microfabrication of these monolasers has a huge impact in the research, especially if you study different biosystems. And it is important to create patterns of these self-assembled monolayers with certain precision. And what people do, they use scanning probe lithography. Here you can see the schematics of this lithography. Usually for self-assembling monolayers, we acquire pictures in a liquid medium. So the substrate, the flat substrate here is gold, and these polymers are attached to this gold surface using thiol group SH, and original surface assessment is done under very low imaging forces. So as you can see that the tip is barely touching the surface here. So very low imaging forces is about 0.05 nanonewtons, so very low. So on the left-hand side here, that is called Nano Shaving, the surface again is characterized under very low force, and then if you apply a bit larger force here, the tip goes all the way to the golden plate, and this is called Nano Shaving, because AFM tip exerts like a very high local pressure on the contact point, and this pressure results in a high shear force during the scan, and this will cause the displacement of everything that was absorbed on the surface of the gold. So this is called nano shaving. And in the middle is nanografting. It is when what we say. So in the medium, you can see a different type of the polymers. So here is just like with a longer tail. So when you conduct this nano shaving procedure, you have a free space left. So these new type of the polymers will be attached to the surface. and or absorbed, and that's how you can basically do lithography, so-called lithography. And on the right hand side, this is another example from the papers. So so here researchers, they were working with thiolated single-stranded DNA. So instead of these polymers, they had DNA with thiol group. And thiolated single-stranded DNA films, they can be self-assembled at a different ionic strength. And again, here they used golden plate to do so. Here they used two different modes. Yeah, we'll explain a bit later about each mode. So here you can see that the ionic strength is different. It's 1 millimolar sodium chloride and 1 molar sodium chloride. And in a shaving experiment, molecules were removed selectively from a certain region from the plate. And here you can see that the thickness of this removed area was a bit different. So this one is from 0 to -4. Again, the reason why it is -4 nanometers is because we were removing the layer. So what we are actually interested is in the difference of the height, not the actual height. So, and because in a low ionic strength, DNA molecules, they tend to be not very, how to say, not very firm, and they do not very perpendicular to the surface. So you can see that the concentration of these strands are much lower in comparison with the experiment when we had high ionic strength. So when you have a lot of ions in a solution, then this allows DNA strands to be more or less perpendicular to the surface that we are immobilizing. So that's why we have larger shift in height, because when we remove scratch all the sequences from here, so the difference, as you can see, about 1 nanometer, 1.5 nanometer, this is the thickness of DNA. But if these DNA stand still, then you can see much higher shift in the height here, because these strands are perpendicular. So this is, so again, so contact mode, even though it kind of destroys the sample, you can use it on your advantage and then destroy the sample in a way that you want it to be destroyed. And for example, here on this free space, you can modify the surface with different molecules. So this can be quite useful. So this is another example of contact mode AFM. So I think you might have some idea that usually contact mode AFM is used to characterize more or less solid substrates. And for soft biological systems, it requires some sort of skill, so you do not destroy your sample. So if you have to use contact mode on the biological samples, usually you would try to avoid large forces. Large force means it should be larger than 100 piconewtons. So everything that is below might work for a sample. So here, this is the comparison of the dynamic mode and contact mode. We will talk about dynamic modes a bit later, but yeah, but this is just as an introduction and the comparison between dynamic and a contact mode. So on this picture, what you can see, So here we were checking the proteins, transmembrane proteins, and here we have a membrane, and then when we go along the membrane, then we have the shift in the height every time when we encounter um, a protein. So ideally, if we go for dynamic mode, the tip touches the sample only when it goes in these points, right? So it is less damaging for the membrane. So these kind of experiments are called like amplitude modulation AFM. So on the picture B, you can see the proteins on the surface. These are potassium channels in the lipid membranes, and on picture C and D, these are another example of different membranes as a rhodopsin dimers, and here you can see that, so these are a bit different resolution. So this is 50 nanometers and this is 15 nanometers. So the feature of the rhodopsin that is very highly packed on the membrane surface. I will talk about rhodopsin again in this lecture. And image E is the cell itself. And here you can see collagen fibrils coating the substrate. So every time when the cell moves, it kind of creates this kind of these features and then gradually moves on the surface. So contact mode can be very useful for imaging the proteins on the high resolution, but what you should keep in mind is that the force should be very, very low. And in the bottom row here, F, G, and H, these are dynamic AFM pictures. So these pictures were done in a contact mode. These ones are in a dynamic mode. So when you go for dynamic mode, the resolution is much higher, as you can imagine here, right? So for example, F, this is the antibody on the surface of the mica, and this is a single protein. This picture is viruses and that are packed in a crystalline assembly. And on the H, you can see a part of a plasmid DNA. So again, what I want to repeat that the higher resolution can be achieved, but usually it is done in a dynamic mode because these molecules are quite fragile. And let's talk about rhodopsin a bit more. So for those who are not very familiar with protein biology, bacterial rhodopsin is a protein from halobacteria, and it acts as a proton pump every time when the light when this protein is exposed to the light energy, and basically it transfers a proton across the membrane. And once it transfers the proton across the membrane, then pH gradient is occurring, and then it is used to generate ATP, and then that's how cells can generate some chemical energy from the light. So bacterial rhodopsin is usually present as a very, very high density protein in the membranes, and because of this high density, it is organized in a crystalline manner. So this is the low resolution scan of the membrane, and this is high resolution scan of the membrane. And as you can see that it is very densely packed, and it forms this kind of triangles. And on the middle part here, this bacterial rhodopsin was measured in a buffer solution. And here you have two membrane patches containing bacterial rhodopsin. And so these curve here is just shows our electrostatic interactions between tip and the surface. The reason why the height is different here, so you see is like about 2 points difference between left and the right, because left patch faces extracellular side up and right patch faces cytoplasmic side up. So this graph shows us the difference in pH, and because protons have charge, this means that what we're measuring here is the electrostatic difference. So that's how you can use contact mode as well. So it's not just about the topography, but it's also about other features such as pH and electrostatics. This is the example of lateral force images. And lateral force mode can be very effective at picking up material difference in the sample. And so, for example, this AFM image is a polymer blend of polystyrene and polybutadiene. Left one shows the topography where two components in the blend, and it's a bit hard to distinguish which polymer is where. So what we can do, we can go for lateral force microscopy, and then now, as you can see, we can differentiate between two polymers on the image. And especially please pay attention to these graphs. So the friction is illustrated in this profile here. So the black profile belongs to one direction of the scan. Gray profile belongs to the opposite direction of the scan. And this is the superimposed image of this scan. And every time when we cross a different polymer, then we have different type of the friction, and that's what is shown on these pictures. So yeah, this is another example of the applications. And now we are going to dynamic or oscillating modes. So there are like huge number of dynamic modes, and however, what I can say, all these modes are just a variation on the topic. So the cantilever is oscillate, and usually it is moved by a piezoelectric element, and usually it's moved at its resonant frequency. So when the oscillating probe approaches the sample surface, the oscillation changes because of the interaction between probe and the sample. And the change of this oscillation is called damping of the cantilever. What does it mean? This leads to a reduction in the frequency and amplitude of the oscillation. So, and then these changes are monitored by the force transducer. And the scanner will adjust the height and via feedback loop control. So the probe will be maintained at a fixed distance from the sample, more or less, if in the same way as we go in a contact mode AFM. So the only real differences between different oscillating modes is the size of the amplitude or of the oscillation that we apply to the probe, and also the difference is in the method that we use to detect the change in this oscillation. So this oscillating signal is generating and it's applied to cantilever mechanically, right? So here you can see the difference between the movements. So when we use small oscillation amplitude is in like red color A, it will be possible to maintain contact AFM, or alternatively what we can say close contact AFM. This technique has some advantages because we have very low forces between tip and sample. And on another hand, if we apply large oscillation, here it's in the blue, the probe will be moving from very, very far position to the where is no tip and surface interaction, and then we'll go through the attractive phase into repulsive phase and back to the oscillation. So this technique involves very large probe and sample forces and can be a bit more disruptive, but it's much easier to implement. So this kind of technique is usually called intermittent contact mode AFM. Okay, so this is just like general introduction, and then now let's go for the equations. So to get some basic understanding of the underlying physics of the dynamic mode, let's just simplify the system. So let's assume that the vibration amplitude is small compared to the range of force interaction. So van der Waals forces range is over 10 nanometers. The vibration amplitude should be less than 1 nanometer. That's what it means. And here we have two springs, and what these springs are explaining here. So the tip will be attached to a cantilever with a spring constant K, and the force interaction will be modeled by a spring stiffness equal to the force gradient. And just also please don't forget that the force interaction spring is not constant, and it will depend on the distance between tip and the sample. Distance is Z here. So the derivative of the force it represents the spring constant KTS here. So this constant will just we will use it to calculate the oscillation cycle. Okay. Also, when we compress this spring into some delta Z distance with its spring constant K, then we can directly measure the force, and this is deformation that can be calculated by Hooke's law. Okay, so we are interested in these two springs, and the two springs, they are coupled in parallel, since the sample and tip both of them support rigidly connected to a certain value Z0. So this is constant, and what is changing is the tip position. So we can write the total spring constant of the AFM system. It is depicted here, total spring, and this is the sum of these two smaller springs. So from the simple harmonic oscillator, when we neglect any damping effects that I was talking about earlier, we can find the resonant frequency omega, and this resonant frequency is the sum of the shift, so difference of this frequency, and the free resonant frequency, omega 0. And this is the actual equation for this, and it depends on this total spring constant, and m here represents the effective mass of the cantilever. So I will not put all the equations here. If you are interested in the total derivation, you can check our textbook for this. So if we approximate that this delta omega is this delta is much smaller than the resonant frequency, so we can write this kind of equation. We can find that the frequency shift of the cantilever resonance is proportional to the force gradient of the tip sample interaction. Even though this consideration is based on a very simple model, it shows us qualitatively that in dynamic mode, we will find that the oscillation frequency depends on the force gradient, and in principle we can calculate the force curve from the force gradient and vice versa. That's why I spent so much time on different curves showing you like last time. However, we neglected quite important issue for AFM so far. So the mechanical stability of the measurement. So in static AFM, the tip approaches the surface very slowly, and then the force between the tip and the surface will always be counteracted by the restoring force of the cantilever. So this is what you can see on this picture. So here again is the typical force distance curve. And as the tip approaches the sample, the negative attractive force that is representing van der Waals or chemical interaction force, these forces will increase until the maximum is reached. So the spring constant of the cantilever here is represented by the slope of the straight line. So the dashed line is the slope over the dashed line is when the force curve becomes larger than the spring constant of the cantilever, and then the tip will suddenly jump to position C from position B. Okay, let's start a bit from the beginning. So when the total force is zero, so this means that cantilever is in its equilibrium position, and so on. So as soon as the position from this equilibrium position A, as soon as we reach it, we find two possible intersection points while we are approaching the surface. So between points A and B, the tip is at its local energy minimum, and then it will follow the force curve. But at point B, when the adhesion force upon the further approach will become larger than the spring restoring force, the tip will suddenly jump to position C. So when retracting the tip, we will pass point C because the tip is still in its local energy minimum, and only in position D the tip will suddenly jump to the point A again because the restoring force will exceed the adhesion. So here we can see that the sudden instability will happen at exactly the point where the slope of adhesion force will exceed the slope of the spring constant. Okay, so this is just the introduction, and now we are going to all these harmonic modes. So the oscillating cantilever has 3 degrees of freedom: the amplitude, the frequency, and the phase difference between excitation and oscillation. So let's consider that the damped-driven harmonic oscillation. So the cantilever is mounted on a piezoelectric element, as you already know, and it will oscillate with the amplitude AD at frequency omega. So this is the overall equation of the oscillation. So we will assume that the cantilever spring obeys Hooke's law, and then we will introduce a friction force that is proportional to the speed of the cantilever motion. So this is this equation. So alpha here denotes the damping coefficient. So this equation is got by Newton's first law, and it shows us the motion of the position at z. So again, we have the distance z, and z from t is the position along this axis while the tip is moving. So these are just a bit of terminology. We will define our resonant frequency as this, the same equation as here, just to remind you. And we will further define the dimensionless quality factor Q. So this is an anti-proportional to the factor to the damping coefficient. So what is this quality factor? Quality factor mainly represents the internal dissipation in the vacuum of the cantilever during these oscillations. And for example, when we have low damping, then we will, and while we're using high quality cantilevers, we will have the change of these Q factor. So for example, how you can see this on the practical experiment, it's a different amplitude here in a different Q factor values and different phase as well. So again, this quality factor will describe the number of oscillation cycles after which the damped oscillation amplitude decays. So after some basic manipulation with these equation, we can get this kind of differential equation. And the solution of it is a linear combination of 2 regimes. So for example, when we start from the rest and switching to the piezo excitation at time 0, the amplitude will increase from 0 to the final magnitude here, and will reach a steady state where the amplitude, phase, and frequency of the oscillation will constant over time. And then the steady state solution, this is Z1, can be reached after 2 quality factor oscillation cycles, and will follow the external excitation with the amplitude A0 and the phase difference phi here. In the transient regime, the oscillation amplitude will look like this. And so the important fact is that the exponential term causes this Z2 to diminish exponentially with the time constant t. In the vacuum conditions, only internal dissipation in the cantilever will be present, and Q will reach the values about 10,000 at typical frequencies, about 100,000 Hertz. So this will result in a relatively low transit regime, which will limit the possible operational modes for dynamic AFM. So that's why the quality factor value is quite important. And if we evaluate this steady state solution as Z1, we can find the solution for the amplitude and phase of the oscillation. And both of these values can be a function of the excitation frequency omega. So these are the equations for the amplitude and phase. The amplitude, again, will reach its maximum at a frequency different from its resonant frequency if the quality factor has almost infinite value. The damping term of the harmonic oscillator will cause the resonant frequency to shift from its resonant frequency to some modified frequency, and this is the equation how we can calculate it. So this shift is negligible for Q factor, which is larger than 100. And this is the case for most applications in vacuum or air. However, if you measure your sample in liquid, this Q factor can be smaller than the 10, and then these resonant frequency will differ a lot from the new resonant frequency that we get. So that's why please pay attention to the Q control factors. Okay, and the next slide just shows us how the amplitude can be changed, and these basically the same graph that I was explaining here. So the resonance will shift to lower frequencies and will show us the drop in the amplitude, and what kind of parameters we can optimize. First of all, we can optimize cantilever spring constant. So the stiffness of the lever must suit the material that you want to image. So if the cantilever is too stiff, the result may be destruction of a sample and wearing out of the tip. If the cantilever is too soft, it may not be able to interact with the sample and generate any contrast. So this is first parameter. Second parameter is the free vibration amplitude. So this amplitude of the oscillation is basically occurs when we have some free space between the cantilever and the sample, and this parameter is set in units of voltage. So rougher samples will need a larger free vibration amplitude. And the third parameter is the set point. So the set point is basically the reduced target amplitude, and it results after the tip is in intermittent contact with the sample. So basically it expresses the percentage of the free vibration amplitude. Lower amplitude set points will be better for aggressive tip sample interaction and more repulsive tip sample interaction. Okay, so this was the theory behind the dynamic mode. And yeah, let's go to the next slide. Dynamic mode as a non-contact mode. So non-contact mode will offer some advantages over contact mode, and it will work via the principle of amplitude modulation detection. Okay, yeah, here we go. So here what we do, we can change the amplitude A and of the oscillation cantilever while it is interacting with the sample. So this change of the amplitude will be used as an input of the non-contact AFM feedback, and to obtain the image we usually set the amplitude as a constant some value, and then and when we put it in such a way that when the cantilever is far from the sample. So the advantage of the non-contact mode is that the tip never contacts the sample, so it will not destroy it. So it's quite important for the biological applications. Next one, dynamic mode as a tapping mode. Here, again, we touch the surface, but not constantly. I kind of tapping it, and here we have oscillation of the probe between attractive and repulsive part of the Lennard-Jones potential. So yeah, so this is the tapping mode, and the next one is the phase contrast. So phase contrast imaging is one of the most common imaging methods, and it is a form of a dynamic mode, and it refers to the phase channel and an excited cantilever oscillation will exhibit the phase shift between the drive and the response, and it can be defined by this equation. So this is the deflection of the cantilever, and it depends on the amplitude, oscillation frequency, time, and the phase shift. At the resonance, the phase shift is 90 degrees in comparison to the phase of the when it's below resonance, and the interaction between an oscillating cantilever and sample changes. The resonance frequency of the cantilever will also shift. So to lower frequencies for attractive forces and to higher frequencies for repulsive forces. And as a consequences, the phase at the fixed frequency shifts when the cantilever sample interaction changes. For example, the material property changes. That's what we see on the phase mode, right? Okay, and so again, as you already know, it is quite useful method for characterization of the polymers and different biological samples. We will talk about these phase modes a bit more on the next lecture. Okay, so this is just a very brief introduction to a dual pass mode. We'll talk about different modes in the next lecture. Technically, it will provide a topographical information of the surface when it goes for the first pass on the blue line here, and it will give us some functional signal when it scans the sample during the second pass when it's not attached to the surface. So in a dual pass topography, again, we're measuring like forward and backward passes, and it will provide more accurate correlation between topography and long-range signal. So basically we are checking the same sample twice. And let's go for some examples how we can use dynamic mode. So here is the example of monitoring different conformations of flexible synthetic polymer molecules in some solid-liquid interface. So these molecules are polypyridine protonated cationic change. So with this abbreviation, I will just call it polymer because it's quite a long name. When it is protonated and in the liquid, it can change its conformation in the medium when we change pH. So here's the pH is 3.8, sorry, 4 and 4.2. So this protonated polymer is a weak hydrophobic polyelectrolyte, and water will be a poor solvent for this polymer. So when it is protonated, it will be soluble in the water and will release counterions. And conformation of these coil configuration will be balanced by column repulsion and short-range van der Waals attraction. So as the degree of protonation decreases, so higher pH, less protons, or when we change the ionic strength of the solution, when it will increase, so the extended polymer coils will undergo the quite sharp phase transition, as you can see here. So this is one of the examples. We can check the conformations of different polymers using, and this one is done in liquid phase, so that's why we use dynamic mode here. Next example. Here we investigating the extension and retraction dynamics of the edges of fibroblasts in the cell culture. And force mapping mode allows the protrusion of the active cell edges to be checked. And we can check its kinetics, its thickness, and different mechanical properties. So here we usually use soft silicon nitride cantilever because it is the cell. And here you can see the deflection image of the regions of the motile fibroblasts. So each curves represent the cross section of these white areas. So what we can see here, we can see that if the edge is stable, the amplitude height is much lower in comparison with the protruding edge, which is here because it's constantly moving. This again, so this is how we can check the motility of the cells. Before explaining what you can see here, just few words about the background of this image. So this is the work of the checking the living hippocampal neurons in the physiological conditions. And the reason why the researchers were interested in the hippocampus, because it is associated with memory processing in mammals, and this structure will degenerate in patients with Alzheimer's disease. And Alzheimer's disease, for those who are not very familiar with it, is a neurodegenerative disease, when the most common case of dementia of older people, and usually it appears when when certain protein is aggregated in the brain, usually it's amyloid B peptides and phospho-tau as well. So why the researchers were deciding to check these neurons using AFM, because the advantage of this AFM that we can check this neuron under physiological condition. And here you can see the growth of the cone of this neuron. So the growth of the cone is associated with the growth of synapse. And this is like the 3D image of this process. On the figure A is an overall visible topographic image. And the 3D image of this topography is under picture D here. So phase and amplitude images show us much more fine resolution. So this is B and C. B is phase shift image, C is amplitude image. As you can see, there are like 4 lines, and these 4 lines are the cross sections and then shown in the image E over here. So profile 1 shows us the incremental increase or in a height from the substrate to the cone, and shows us the height of the cone. Profile 3 shows us the phyllopodia. So phyllopodia is these short growing things here, and the height is ranging from like 50 to 100 nanometers or so. So the main idea of this slide is just to show you that even though the topographic image is not very clear, because again, we need to image it in the physiological conditions. So the cell is moving, the liquid is moving, everything is moving. But if we go for phase shift or amplitude images, then we can achieve much higher resolution. Okay, and then just as a reminder, once again, Hooke's law will give us a possibility to look into different molecular properties. And these are how these force distance curves work. And what we're interested in, you should be familiar with the actual image here, is the difference between these 2 angles. That's what we're interested in. And the difference in these 2 different angles will show us the different deflection of the cantilever, and we can measure like modulus, adhesion, unfolding and stretching measurements just knowing the difference of these angles. Oops, sorry. And as an example is here, so we are doing the unfolding and stretching measurements of bacterial rhodopsin. I've already mentioned it today. Bacterial rhodopsin can be unfolded using AFM. So AFM tip is attached to one of the domains here, and each peak corresponds to unfolding moment of each of the domains, and these colors are the same as the colors of each domain here. Mechanical unfolding will occur when we retract the cantilever at the constant velocity v. So each of these transmembrane helix will go out from the membrane, and that's how it will be unfolded. On the right hand side again, so we can see the difference of the force that is happening every time when the final unfolding is actually happening. It's very popular in biological research because very often we're interested how exactly the proteins are folding or unfolding, especially for transmembrane proteins, because it's very hard to crystallize them outside of the membrane and make a structure like a crystallography structure. The example of force distance curves in multi-parametric imaging. So there's always a question if AFM can do something more than just topography. Here we can see the different types of force distance curves. So on the left-hand side, you can see the retraction force distance curves are following the adhesion forces. And that's how we are able to characterize quite reliably the properties of the sample, just by precisely controlling the interaction between tip and the sample. On the picture B here, you can see the pixel by pixel basically pixel by pixel resolution of the sample, and then in each point we can get these force distance curves, and that's how the mapping is done. So each force distance curve locally will quantify different physical properties and interaction, and then we will map it to the overall image here. So these are all the examples for today.
С вами был Тибериус. Если видео понравилось, ставьте лайк. Если видео не понравилось, так уж и быть, ставьте дизлайк. Но обязательно напишите почему. Подписывайтесь на канал. Всем пока. До скорого.